Well, I have to say, I've never had an introduction like that, and it's wonderful. So first of all, I have to say, it's an incredible thrill for me to be here in this beautiful city that I love, and in this extraordinary venue, isn't it? I mean, this is just an incredible place. And thanks so much to Jay Armas and Novo Nordisk for making this possible, and for all of you for coming to listen to this story that I'm going to tell you. I'm going to tell you a mystery story, and I'm going to, hopefully, you'll help me solve it along the way. So, let's talk about the decline in reproductive function. A paper came out of Rikshospital in 1992 stating there's been a genuine decline in semen quality over the past 50 years. That's huge, and actually the decline was about 50%. I was concerned about it. Many people were concerned about it. Let's take a look at the graph from that paper. So what you see here is a decline, actually 1938 to 1991, of about 50% in less than 50 years of sperm count, which is one marker of semen quality. And it doesn't perhaps look so impressive. There are a lot of circles. Those are weighted with the size of the studies. But maybe it isn't real. And this is what many people raised, including me, asking what else could cause this decline. Now, unfortunately, I can't see you because there's so many lights, but I hope you can call out to me if you have suggestions as to things that could artificially produce this decline. What could produce this decline other than true biological change? Anybody have any ideas? Phthalates. You're jumping to the answer. Okay, we don't know that much then, in 1992. What else might be causing the decline? Sorry? Alcohol, that's a good guess. Maybe men are drinking more recently. What else could have happened over time? Pesticides. What else? Plastics. How about how sperm are counted? Maybe the method of counting sperm changed, right? So that sperm counted out there in 1990 were counted using a different method than back in 1938. That's very plausible, isn't it? Or maybe the men were older. Well, sperm count goes down with age, and so on and so forth. So we looked at these different factors, and I asked, if you account for all of these, okay, in a model, using the data from the underlying studies, could you make the slope go away? Could you straighten the line? Okay? So this is the first step of the mystery. Can we make the decline go away? And the answer was, I can give you the answer here, no. After I did this extensive analysis, and actually a second extensive analysis, and considered all of these factors, including geography. I mentioned that because we're going to come back to that, and because this graph you see has 3 lines now: North America, Europe, and other. And you can see that other doesn't contribute much, right? In fact, most of the information comes from North America and Europe. But the key here is that the slope, minus 0.94 million per milliliter per year, was the same, basically, in the 1992 paper and in this paper. I've never seen anything like this. I've never seen a study where after you adjust for confounding factors like these, you can't alter the results. That's how solid they were. And so I had to take it seriously, which I had not, by the way, before. So if it's real, what is causing it? So we talked about artifacts, age, alcohol, sperm counting methods, maybe recruitment methods. But now I'm going to talk about causal factors, things that actually might have caused the sperm count to change. So if you talk about cause, very crudely, you can talk about genetics and environment. That's the first subdivision, if you will. Genetics is not likely to be playing a role here. It's too fast. It's 2 generations, 50 years. We're not going to get a 50% decline in 50 years. Think about if this was IQ. A 50% decline in IQ from 100 to 50? I think people would notice that, wouldn't they? But so why didn't they notice this? You know? Anyway, it's not genetic. We can leave that aside, and that takes us to environment. Environment is very broad, everything that's not genetics. I then break it down into lifestyle and chemical. Again, a broad classification. And I'm going to spend most of my time with you today talking about the chemical, but we'll come back to the lifestyle. So now, if we look at the chemical, I'm going to break that further into 2 categories. One is the chemicals that we call endocrine disrupting chemicals. These are chemicals that the body can confuse with our natural hormones. Hormone mimics, if you will, chemicals that can interrupt the production, transport, or function of the body's natural hormones. And then there's everything else. The reason this is important is because we're talking about sperm count, and that's a reproductive function, and that depends heavily on hormones, particularly the steroid hormones. So that's where I'm going to spend most of our time today. And this mystery then is, among these chemicals, are there some that could be driving the decline in sperm count? Okay, so that's our job now for the next little while. And I will mention the other chemicals at the end, but that's not what I'm going to spend most of the time on. So in order to answer the question of why sperm count could differ over time, I thought, let's address it indirectly and ask, can sperm count differ by place? Okay? I can't really get my hands on the environment in the past, but I could look at different environments today. And so I designed this study, which is the Study for Future Families, using 4 locations in the United States. And by the way, my colleagues at Icahn Hospital also did this in 4 cities in Europe. So we were all after this, on this quest to find out what is it in the environment that could be driving or affecting sperm count. Okay, so I picked these locations for various reasons, but what you need to know is that Columbia, Missouri, which is blue, is different than the other ones. Columbia, Missouri is semi-rural, it's agricultural, and the others are urban centers, New York, Minneapolis, and Los Angeles. And so that's where I wanted to do the study. Who was I going to study? Well, an important thing is that the men that come in these places are comparable. Otherwise, how can you compare them, right? And so what we decided was we would look at partners of pregnant women. Why? Because pretty much every pregnant woman comes into a clinical center for prenatal care. So we have a captured population. And then, if their partners will agree to participate, we have a fairly representative sample of men and women who are who are pregnant, basically. So that's what we decided to do. And then the other thing we worried a lot about was, is sperm count going to be measured in the same way everywhere? Okay? You can understand that if you saw a difference between two centers, but you weren't absolutely sure that the methods were the same, you wouldn't know what the difference was due to. So to ensure that, we had very, very high quality control. All the instrumentation was, the same. Everybody was trained at the same place in UC Davis. There were monthly circulations of semen samples to be measured by all the technicians, and this was a really clean study. And what did we find? We found that in that rural agricultural center in Columbia, Missouri, sperm count was actually a lot lower. In fact, there were half as many moving sperm in central Missouri as in the urban centers, particularly Minneapolis. And by the way, over there on the right, normal sperm, percent normal sperm, that's the shape, that wasn't affected. But the motion and the number were very different in Missouri and in the urban centers. Okay, now let's stop for a minute and think about this. We're looking for an explanation for differences in semen quality. What do you think could be different about Columbia, Missouri, and the rest of the centers that might affect sperm count? Ideas? Agriculture. Great. I love that answer because I have a slide for that right there. Yes. That's the key difference between these centers. And so we wanted to dive in and make sure that the use of agriculture, and particularly agricultural pesticides, were directly linked to the semen quality. Now, I didn't actually have a lot of money for this, but I did have a little, and so we did a pilot study, and we wanted to look at pesticides in the men and relate that to their semen quality. And we could do that because, turns out, we had collected urine from the men. We also collected urine from the women, by the way, which will come out to be useful in a few minutes. So we had this urine, we sent it to the CDC, they measured pesticides, and we only did it for 50 men in Missouri and in Minneapolis because we didn't have a lot of money. So we chose men with every parameter terrible versus semen, and every parameter very good in both places. And this is what we found. That for 6 of the 8 pesticides we measured, the men who had poor semen quality had significantly higher levels than men who had good semen quality. This is pretty convincing, isn't it? I'm convinced. I was convinced, and I am convinced, based on quite a small sample, to have these very significant differences. And so this says that, however, these were not farmers, by the way. These were just people who lived around the clinical center. So this was coming in their air and their water and their food. They were responding to the presence of these pesticides in their environment. And this was lowering their sperm count. So we've now found one factor that can affect semen quality. That's environmental, but it can't be the whole story. So going beyond pesticides now. Okay, I mentioned that there were other endocrine disrupting chemicals, and I just wanted to show you this picture of because I think it's a nice cartoon. It illustrates how these endocrine disruptors in green are like spam, interfering with the message that's being sent. They come in and they hog those receptors that should be occupied by the orange hormones. That's what's happening with these chemicals very superficially, but there are many of them. There are many, many endocrine disruptors that have this kind of property. They can do other bad things as well. And the one that I'm most concerned about for our story today is testosterone. Why? Because testosterone is a critical male hormone, a steroid hormone that's important for the development of sperm. Right? Make sense? So testosterone is what I'm going to focus on now. So here's a nice cartoon that tells us about the role of testosterone in the development of the sex organs. So in this story, originally the sex organs are undifferentiated. They're the same in genetic males and females. This is very early in pregnancy. Then testosterone is introduced or not, and if it is, then the male develops in a genetically typical male way. For example, here he develops testes, and if testosterone is not present, then the female goes on to develop ovaries. So testosterone is very key to this step here. And interestingly, if you look at a rat on the left, a pregnant rat who is not exposed to an anti-androgen or a testosterone-lowering drug, then in her, the testosterone will surge in her pregnancy, approximately days 18 to 21, very specific. But if she is exposed to a phthalate, and this is particularly, this study was diethylhexyl phthalate, which is probably the worst actor, then look, no testosterone surge. Now, if you think about the prior slide I showed you, where that testosterone surge is needed for differentiation of the gonads and differentiation of the genital tract, that's not going to happen if this surge is completely suppressed. So now you should be starting to get convinced that testosterone is important, and this slide should clue you in that phthalates may play an important role in messing up this transition. Okay? So where do we go from here? Well, a very important study by Paul Foster in 2000 said that when the mother was exposed to, this was a different phthalate, also an anti-androgenic phthalate, dibutyl phthalate. When the mother is exposed to this phthalate, then the male had decreased fertility and sperm count. Okay, now we're getting closer to our goal of finding something other than pesticides that can affect sperm count, the anti-androgenic phthalates. But it turns out that this exposure does more than this. It actually produces a whole syndrome, which has been called the phthalate syndrome. Now, this may not sound remarkable to you, but if you worked in this field, you would sort of sit up and take notice, because there aren't very many syndromes named after chemicals. There's fetal alcohol syndrome, and there's the phthalate syndrome. This is a really important syndrome. And it has these outcomes: a smaller penis, a possibly malformed penis, undescended testicles, and the last thing, shortened anogenital distance, is what's going to be key to the next stage of our story. And I looked in and saw that you were seeing something about ABC. Okay, ABC is slang in the US for ass ball connector. It's also known as the taint, the gooch, and the grundle, okay? But these are things that kids know on the street. Adults don't know what the anogenital distance is. Now you will. And this distance turns out to be really, really critical. And it's shortened by phthalates. It's part of the phthalate syndrome. So here's a picture of it, okay? Male, female, rodent. Looks pretty easy to measure. Actually, not so easy because they're pretty teeny. But it goes from the anus to the genitals, AGD. In the female, it's half, 50 to 100% bigger in the male than in the female. And this is true of most mammals. Not all mammals, but most mammals. And it's true in humans. And it should be true in the absence of phthalates. However, the phthalate syndrome, as we saw, includes a shortened anogenital distance. That means that when the mother is exposed to phthalates, her male offspring have a AGD which is not as long as it should be. It's more feminized, if you will, less male typical. Okay? So when I heard about this, I was actually flying on a plane with a colleague from CDC, and he said, Shanna, you should look at phthalates. I said, why should I? I'd never heard of phthalates, actually, to tell you the truth. And he said, well, first of all, they're in everyone in the United States. We measure them at the CDC now pretty inexpensively, and we've shown that. And they cause, I'm using that word advisedly, they cause the phthalate syndrome. Okay? So I was worried about declining sperm count. And I thought, maybe this is going to give me a path. Maybe this is how I'm going to solve the mystery of why sperm count is declining. And so I asked, could there be a phthalate syndrome in humans? Okay? So think about how you would answer that question. Well, you need to look inside the fetal environment. Well, we can't quite do that. But what we can do is look inside the mother's body via her urine. So it turns out that phthalates are water soluble, okay? And so if she's exposed to phthalates, this shows up in her urine, and you can measure the urinary phthalate metabolite levels. And once you do that, you know how much phthalate she was exposed to. And then you can look at her baby. And in her baby, you could, in principle, if you knew how, look at these measurements. Now, penile width is not rocket science. That's pretty easy to measure. Testicular descent has long been known, you know. But AGD was a little trickier. And so designing the study that would measure AGD took some time. And what babies were we going to look at? Well, remember the study for future families that we looked at for the 4 cities in the United States? Those women had given us a urine sample, and they were pregnant. So we knew what phthalates were in their bodies, and then we could bring their babies back, and we could look at these outcomes. So, Study for Future Families became the study of phthalates in pregnant women and children, and we measured their anogenital distance. This is how we did it. It's not rocket science, it's pretty easy. It's easy to do repeatably, reliably, accurately. So, what did we find? Well, this is just a snapshot of results from that study, but it's kind of, will give you the picture. So if you look at the metabolite MEHHP, one of the metabolites of diethyl hexylphthalate, which is a big molecule, has a lot of metabolites, it ranges all the way from close to 0 to very high, but the 75th percentile is 20 nanograms per milliliter, and that's, we'll call that high, and 25th percentile is 6. So if you compare the AGD for a mother who's in the upper quartile versus a mother in the lowest quartile, you get the figure on the right. She is 13 times as likely to have a son with a shorter than average AGD. 13 times. This is huge. And compared to a mother under the 25th percentile. And if she's in the middle at the median, then she has a almost 5 times greater risk of having a boy with a short AGD compared to mothers in the lower quartile. So this was a very, very strong association between the mother's phthalate levels and the boy's anogenital distance. Once we'd done this, we really had to do it again. And we did it again by designing a second study, and this study was really designed specifically to look for the phthalate syndrome in humans. The other one was a sort of a tag-on to an existing study. Things were not quite right. The urine was everywhere in pregnancy. The babies were measured not exactly at the same age, but in this one, we did it right. And we found it again. And so the summary is that we confirmed the hypothesis of a phthalate syndrome in humans in these 2 publications, and we showed that phthalates were endocrine disruptors, and we showed there was a phthalate syndrome, and that AGD was a marker of fetal testosterone exposure. This took a long time, and this took millions of dollars. And I just mention that because this is identifying the action of one class of endocrine disruptors on one class of outcomes. And when we now blow this out to other endocrine disruptors and other outcomes, you can see to have this kind of proof is prohibitive. We can't keep doing this. We can't keep putting these things into our bodies and having to show that they're harmful. It just takes too long and it costs too much money. So we have a short AGD linked to phthalates, but this does not directly link to sperm count and fertility in these boys. These are babies. They didn't have a sperm count that we could measure. They didn't have evidence of fertility. So what we had to do was to go back to that animal study where we said, okay, in rodents, these phthalates are related to low sperm count and infertility, and a study in rodents that said, if you're born with a short AGD, it's yours for life. You're stuck with it. And once we put those 2 things together, we decided to go and ask in young adult men, how does their AGD relate to their sperm count? Okay? That make sense? That was the Rochester Young Men study. And what we found was that the shorter the AGD, the lower the sperm count in these young men. Now, we don't know they were born with a short AGD, so this is a missing link to the story, but it's plausible given how the animal and human data has matched up for everything else. And a colleague in California, Mike Eisenberg, looked at it in fertile versus infertile men and showed that infertility was associated with a short AGD. It's also associated with male genital malformations. So it seems to be a pretty good marker of disruption of development in early pregnancy. Let's go back to sperm count. That study from 1992 was pretty old by the time we got here, and we asked, well, has it got better? What's happened? And the way to answer that is really to do another search of the literature. So here's our team. I think Niels Jorgensen, who's up there on the top left, is in the audience today, I hope so. He was an important member of the team, and we had other andrologists and statisticians, epidemiologists, and a reference librarian. And we decided that we were going to do the most complete, the most thorough study of sperm count in published literature that had ever been done. And it was huge. Just running through this quickly, we looked for English language studies. We found 7,500. That's a lot of studies to read, isn't it? But we were able to throw out about 5,000 of them quickly based on abstract and title, because they were clearly not what we were looking for. They might have been animal studies, they might have been studies not with primary data, and so on. So that's good, we got rid of that, 2/3, and that left us with 2,500. That's still a lot of studies to read in detail, and that's why we had such a big team and took so long. And we did that, and we winnowed them down to 185 studies. Now, I just remind you of the numbers. Carlson started with 61. My reanalysis had 101. Now we have 185. So in these 185 studies, we did an analysis using contemporary methods, meta-analysis methods, which hadn't been available earlier, and asked the question, has sperm count declined? This is what the studies showed us in terms of numbers. First of all, I have to say we divided them geographically again, as I had before, and this time into Western and non-Western studies. That is Europe, United States, and Australia. New Zealand were the Western studies, and then there was the other studies. And then we divided them into two groups based on their fertility status. Studies of fertile men and studies what we call unselected men, that is men who had no knowledge of their fertility. And those are the men that are going to be most representative of the population. They have no vested interest in participating in a study. They don't want to find out, is it good, is it bad? They're just neutral, if you will. College students, military recruits, and so on. And we ended up with a mean sperm count of 81 million per milliliter, total sperm count, which is the concentration times the volume, of 260 million. And this is what we found. If you look at those four groups, unselected, North America, Australia, and New Zealand, and the rest of the world, AAS, that's Asia, Africa, and South America, divided into unselected and fertile. The most important group here is the blue line. That's the unselected men from Western countries. That's our strongest finding. And that finding is completely consistent with the prior publications that I showed you, the Carlson and my reanalysis. And the conclusion was that sperm count had declined 52% in 38 years. That's something over 1% per year. Very, very consistent with the earlier studies. And this made big, big news. Here's a nice headline from Newsweek, and it led to an agent coming to me and said, "Would you write a book about this?" I don't think so, but eventually yes. And so that's led to my writing Countdown, which is the book that I'll show you the jacket of at the end. Anyway, there are other reproductive outcomes, aren't there? Besides men, there are women. Besides sperm count, there are things like testosterone, and there are other causes besides those that I've talked about. I'd like to just briefly go through some of those. So this diagram, this kind of Venn diagram, is nice because it shows you reproductive problems in men, in women, and shared, or those that both have. And we've talked about sperm count, and we've talked about short AGD, smaller penises, not so much about reduced testosterone, which has gone down worldwide at about the same rate as sperm count. Genital birth defects have gone up at approximately the same rate. These rates are about 1% per year. And on the female side, low birth weight, that's a pregnancy outcome, menstrual problems, endometriosis, premature egg depletion, which is premature ovarian failure, not enough eggs left as you approach menopause. Early puberty, miscarriage, going up at about the same rate. And perhaps most dramatically, we have the decline in fertility. So fertility is complicated. This is the fertility rate. This is the number of children born to a couple or a woman in their lifetime, expected or actual. And we need about 2.1 children per couple to replace ourselves. And this decline is, again, 50% in 50 years approximately. And in some countries, particularly the Asian countries, it's actually quite a bit below 2.1. It's all the way down to 1.0 in some countries. So this is definitely going down, but it isn't only environmental chemicals. So let's go back to this graph or schematic, and I'm going to now say something about this other box here that I said I'd come back to. This is the lifestyle factors. So these are things like your diet, whether you eat a Mediterranean diet or you eat a Western typical diet, binge drinking, stress, heat, obesity, couch potato behavior. All of these factors are important in both sperm count and fertility. And many researchers here in Icahn Hospital and in the universities I work with in the United States have done many, many, many studies on each of these factors, and there's no question that they all play a role in both sperm count and fertility. However, many of the sperm count studies and many of the other reproductive studies control for these. So these are usually things that we try to control for. But there are also things that you might think about if you are having trouble maybe having a baby. These things are playing a role. There are also things that are important for other health conditions like heart disease and so on. These are not unique to reproductive function. And let's just briefly talk about the other EDCs. So I talked about phthalates and I talked about pesticides. The bisphenols are chemicals that actually increase estrogen. So phthalates lower testosterone, the bisphenols increase estrogen. Phthalates make plastics soft and flexible, bisphenol make plastics hard. So hard water bottles, baby bottles, Those are the bisphenols. Bisphenol A, could also be bisphenol F, bisphenol S. Then there are the PFAS chemicals. Those are the barrier chemicals, if you will, the chemicals that are lining your nonstick pans, that are in your your water-resistant jacket, and the paper on your pizza box that keeps the grease from going through are some examples. And the flame retardants are chemicals in our living room, in our floors, in our carpets, in our sometimes children's clothes. Let's look at what's in our house. So we can start with our kitchen, and I want to point out that I love this. This is an actual ad recommending that plastics be made an important part of your healthy diet. And I wouldn't agree with the headline, but they are an important part of our diet. That is, they contribute importantly to our diet, not in a positive way. And, oops, I skipped the, okay. So what I want to show you here is that eating organic, which you in Denmark are close to doing universally, I think, here, is going to keep pesticides out of your diet, and also the chemicals that accompany pesticides. For example, phthalates are put into pesticides because they increase absorption. So by eating organic, you're not only getting rid of pesticides, you're getting rid of other bad actors as well. And by eating dairy that's free of hormones and that's fed on organic feed, you're also keeping these chemicals out of your diet. Once the food gets into your house, it's your responsibility to keep it free of these chemicals. And I think everybody knows that you don't want to store food in plastic. You don't want to microwave food in plastic. Phthalates, for example, are not chemically bound to the plastic. They can leave it when it's heated, and so they go into the food and then into our bodies. And these things are found in the playroom, they're found in the living room, they're found in the bathroom, in your cosmetics, in your personal care products, in your shower curtain, and they're found in your garden, in your PVC containing hoses, and in the pesticides you spray. So they're really everywhere. And I want to just, I'm close to ending here, and before I do, I want to just mention that we're not alone on this planet, and other species are being exposed to the same chemicals that we are, and not surprisingly, they're having similar effects. So there's many studies of measured levels in these animals associated with poor reproductive function.
This is really scary information, and I'm wondering why this not, why isn't this not on the news first story? I don't understand why you have to come to some meatheads podcast to explain this. Because I'm like, how is this not being picked up by major news organizations?
Epidemiologists have found humans across the globe are becoming increasingly less fertile.
New medical findings suggest that in the not-so-distant future, most men could be infertile.
59% drop in total number of sperm in a sample between 1973 and 2011.
Research shows the worldwide fertility rate dropped by nearly 60% between 1960 and 2018.
But we're changing human beings.
Absolutely.
Like what it means to be a human being,
That's
a developing human being is a different thing now.
Right.
Because of poisons.
Right.
Wow. Why are they not sounding the alarms? We need to figure this out, and we need to figure it out now.
Thank you very much. I hope you read more and Countdown, and I hope you have a lot of questions for me. Thank you.